Good afternoon. Thank you so much for the invitation to talk today. I'm excited to talk to you all about a topic that I find really interesting as someone who practices both pediatric pathology and molecular pathology, which is the role that variants that we typically think of as oncogenic play in malformations and other developmental disorders. I have no conflicts of interest to disclose. So today we're going to start out by reviewing some of the roles that oncogenic variants can play in vascular malformations and some other somatic mosaic lesions, with a focus specifically on KRAS variants and kind of the phenotypic heterogeneity that you can see with those. And then we're going to move into another topic that we more typically associate with cancer, which is genetic complexity and what we know about the role that that plays in vascular anomalies. I think most of us in this room are familiar with classic germline inheritance, wherein you inherit a variant from one of your parents, and that is then present in every cell in your body. And some classic examples would be things like cystic fibrosis with autosomal recessive inheritance, or Li-Fraumeni syndrome with autosomal dominant inheritance of a predisposition to a variety of cancers. However, it's become obvious over the past several decades that mosaicism also plays a large role in human disease. And mosaicism is defined by the presence of two or more cell lines that are derived from a single zygote, meaning that within a single individual, you'll have both wild-type cells as well as cells that are carrying a mutation. And the idea here is that there's some sort of error that occurs during DNA replication, during mitosis. And that error is then passed on to every cell that's derived from that initial event. But before we talk about mosaicism and disease, I think it's important that we take a moment to recognize all the physiologic roles in which mosaicism is important. And one of those would be our immunoglobulin and T cell receptor rearrangements. So all of our B and T cells have a variety of different immunoglobulins and T cell receptors that they express to help protect us from infection. In those of us that have two X chromosomes, there's random X inactivation within different cells. I studied telomeres for my PhD, so I'd be remiss if I didn't briefly mention those. But every cell in our body has a different telomere length, depending upon the number of cell divisions that cell has undergone and the activity of telomerase within that cell. And then moving from our main genome to our mitochondrial genome, there's actually this phenomenon also called heteroplasmy, wherein most of us have different subpopulations of mitochondria within our cells. And lastly, in addition to all of these physiologic roles that mosaicism play, all of us are sitting here right now having variants introduced into our DNA as our cells divide and our DNA replicates. And that error rate can be as high as 1 nucleotide per billion nucleotides within the retina, which sounds really low until you consider the fact that the typical diploid human genome has 6 billion nucleotides. So that's actually a pretty high error rate given the size of our genome. And there's a few different large-scale studies currently ongoing to really classify the spectrum of mosaicism within an average adult. But this paper from 2010 did a nice job of trying to estimate what it would be. And so in your average, normally developed human adult, you have about 10 quadrillion single base substitutions in your genome that presumably were not there at the time when you were born. So we're all accumulating these over time. Now this figure here is a little complex at first glance, but it depicts the different outcomes that you can get from a mosaic variant when it occurs in an adult. Let me get my laser pointer going here. So the y-axis here is the clone prevalence, and the x-axis is time. And you'll see this F, which is the fitness of a cell that carries that mutation in comparison to a wild-type cell. So for example, just by chance, we would expect that most of these mosaic variants would occur within non-coding regions of the genome, and those would be expected to have no effect on cell fitness. In contrast, some mutations will have result in a decrease of fitness, and those cells will be outcompeted by wild-type cells within that individual, and they can result in either cell cycle arrest or apoptosis, or those alterations can sometimes undergo reversion. And as pathologists, where we probably see mosaicism the most often is when it results in some sort of detectable disease. And those are most often those mosaic variants that result in a gain of cell fitness and give that cell some sort of advantage over wild-type cells. So here I've altered this figure somewhat to think instead about mosaic variants that occur in utero. And when they occur in utero, there's a few outcomes that are somewhat different than what we think about in adults. One of those is that you can have variants that result in miscarriage, and in some individuals, detectable mosaicism can manifest as malformations. So, for example, again, just like adults, many of these variants would be expected to have no effect on cell fitness and not result in a detectable phenotype. Others might actually result in a decrease in fitness, and those are the variants that can result in miscarriage or might be tolerated within one cellular compartment, like perhaps they're tolerated in the placenta, but not in the developing fetus. And what we're going to spend a lot of time talking about today are those mosaic variants that occur in utero and result in an increase in fitness. And one of the potential outcomes when you have an increase in fitness is developing neoplasia, similar to what you see in adults. But a unique situation that occurs in development is that you can also develop malformations because of these mosaic variants. And so the idea here is that mosaic mutations that occur early in development lead to malformations because they in some way alter the primary developmental program within that cell. So shown here is an example of a congenital pulmonary airway malformation, where we think that a KRAS mutation occurs really early on in lung development, probably during early lung budding. And it's from that initial KRAS mutation that the malformation is then derived. And we now know that somatic mosaicism plays a role in many different pediatric diseases, working our way kind of from the head downward. Focal cortical dysplasia is one of the common causes of pediatric epilepsy and is caused by mosaic variants predominantly in mTOR, but also in PIK3CA, AKT, and other genes. Congenital pulmonary airway malformations are predominantly caused by variants in KRAS, sometimes rarely in other genes as well. We'll spend a lot of time talking about vascular malformations today, but the most commonly implicated gene in those is PIK3CA. Additionally, many of our patients who have congenital nevi, whether they're congenital melanocytic nevi, nevus comedonicus, or sebaceous or epidermal nevi, those are also mosaic lesions. And lastly, in patients that have localized overgrowth, that often has a mosaic cause, whether it's mosaic Beckwith-Wiedemann or mosaic PIK3CA variant or mosaic variants in some other genes we'll talk about today. But the critical thing to remember about these lesions is what I find most interesting about them is that they're often caused by the same highly activating oncogenic variants that cause adult-type cancers. But when these mutations instead occur in utero, they result in malformations instead of neoplasia. And so in my practice at Seattle Children's, by far the most common mosaic disorder I see are vascular malformations. So we're going to spend some time diving into those. And the International Society for the Study of Vascular Anomalies, or ISVA, has published a classification system for these lesions. It was just updated in 2025, and you can go on their website and take a deep dive into the classification system if you would like. But at the most basic level, this classification system divides vascular anomalies into one of two groups. The first group is that of vascular tumors. These are lesions that generally have kind of an expansile growth pattern, and they can occur at any point during a patient's life. They can also grow disproportionately to your patient, or they may regress over time. And it includes everything ranging from benign hemangiomas, like this lesion here, which is an infantile hemangioma, to borderline lesions, like hemangioendotheliomas, to frankly malignant angiosarcomas. In contrast, the other major branch point here is that of vascular malformations. And vascular malformations are lesions that our patients are typically born with, and they're going to grow with our patients over their lifespan. This here is an example of a lymphatic malformation within the parotid gland. And because these malformations have been present since that organ initially developed, you'll often see that it kind of intermixes with the background normal tissues. So here you can see those kind of malformed lymphatic channels intermixing between these preserved islands of salivary gland tissue. And vascular malformations are typically divided up by the type of vessel involved, whether that's lymphatic, venous, capillary, arteriovenous, or any combination thereof. And the majority of these vascular malformations are somatic mosaic lesions. And so the idea is that at some point, very, very early in development of that vessel, whether it's within some sort of endothelial cell precursor, within a stem cell niche, or an early blood vessel in development, you have a mutation that occurs. And it's from that mutational event that the vascular malformation then arises. And so there are a lot of different genes that have been implicated in vascular malformations and vascular tumors, many of which I've highlighted here in blue. But the critical thing to know about these different genes is they have a tendency to converge on one of two different pathways. The first is the PIK3CA-AKT-mTOR pathway, and the second is our RAS-RAF-MAP kinase signaling pathway. And ultimately, the downstream effect of activation of these pathways is that you get increased cellular differentiation, proliferation, and angiogenesis within these lesions. And at Seattle Children's, to help care for our patients that have vascular anomalies, we developed what we call our VAN-seq panel, which is a targeted capture NGS panel. This was developed in 2019, so I can take no credit for this development, but it was really Jimmy Bennett and Candace Myers who did a lot of work on this together with others. And using this panel, we sequence targets to a really high depth, which allows us to detect variants down to about 1% variant allele fraction. We'll talk about why that's important in a moment. And in its current iteration, it includes these 47 genes listed here, as well as a variety of subpanels, and we accept many different tissue types, including FFPE. And this test has been very successful, so we routinely sequence patients from all over the United States and Canada, and have sequenced patients from a total of 5 different continents. And in 2025, we sequenced almost 600 individuals that have these rare disorders. In total, since 2019, we've sequenced about 2,100 individuals with suspected somatic mosaic disorders, and this includes predominantly patients that have either vascular malformations or overgrowth. And we've detected a pathogenic or likely pathogenic variant in about 2/3 of individuals that we've sequenced. As you would expect for a mosaic disorder, the diagnostic yield is substantially higher for tissue specimens than it is for blood or saliva. And when we look specifically at our tissue samples, our diagnostic yield is around 70%. So in 7 out of 10 individuals, we're able to find a genetic cause for their disorder. This graph on the right depicts the variant allele fraction of the variants that we've reported. And so variant allele fraction is related to the number of total DNA molecules that carry a specific variant. And in this case, it's binned off of 2.5% bins down here at the bottom. And what you'll notice is that almost half of our cases have a variant allele fraction below 5%. And actually 17% of individuals that we sequenced have a variant allele fraction below 2.5%. And so that's why that really high sensitivity is really necessary for these lesions, because it's a very small proportion of the total cells in the tissue that are carrying the pathogenic variant. This graph here depicts the different genes that we've reported variants in. And as I mentioned, PIK3CA is by far the most common. And that gene alone accounts for about 37% of our patients. But there's a variety of other genes that we find variants in as well. And these kind of top 5 genes here account for almost 3/4 of patients. And amongst those top 5 genes, I think PIK3CA and KRAS really both stand out because they're both well-characterized oncogenes that I think we all know very well from the cancer realm, but they can result in a variety of different phenotypes when they're instead mutated in utero. So we're going to spend a moment talking about PIK3CA, and then we're really going to dive into KRAS. And PIK3CA variants can be found in a variety of different mosaic lesions, including focal cortical dysplasia, vascular malformations that are typically slow flow, so that means they're either lymphatic or venous or capillary. It can also be found within epidermal nevi or in patients that have localized overgrowth. And this heterogeneous group of phenotypes that you can get with mosaic PIK3CA variants is called PIK3CA-related overgrowth spectrum disorder. And so this is a group of clinically recognizable conditions that are typically diagnosed either at birth or early during childhood, and includes this really broad phenotypic spectrum. So it includes things like isolated vascular malformations, perhaps a patient that has just a large cystic lymphatic malformation in the neck. It includes fibroadipose vascular anomalies. It also includes our patients that have isolated overgrowth, whether that's an overgrown limb, or sometimes a patient will just have a little overgrown nodule on their hand that's also caused by PIK3CA, to much more complex or severe phenotypes. Like in this picture here, this patient has a diagnosis of CLOVES, which is characterized by overgrowth together with vascular malformations, epidermal nevi, and skeletal and spinal defects. And this is a paper from a couple of years ago now from WashU that characterized their cohort of patients with mosaic PIK3CA variants. The first author here is Bahara Mujarrad, who also happens to be one of my new colleagues at Seattle Children's. But I like this paper because they went through and compared their patients with mosaic disorders and PIK3CA variants, shown here on the bottom, to the spectrum of variants seen in cancer. And what you'll notice is that there are similar trends in both. So in both cancer and mosaic disorders, amino acids 542, 545, and 1047 are the most frequently altered. And so in these patients with lymphatic malformations, we're seeing the same PIK3CA variants that you may see in a breast cancer. All right, so now we're going to dive into KRAS and the many different phenotypes that it can cause. So KRAS is really a central component of that RAS-RAF MAP kinase signaling pathway. And it's a GTPase, and when it's activated, it results in activation of these downstream signaling factors. And mosaic KRAS variants have been implicated in a variety of different phenotypes, including type 1 and 3 congenital pulmonary airway malformations, vascular malformations that are typically high-flow arteriovenous malformations, vascular tumors, and specifically pyogenic granulomas, a variety of different nevi, including both sebaceous and epidermal, as well as in some patients that have sort of a unique overgrowth and vascular malformations phenotype that we'll talk about. And shown here is a pie chart of the different KRAS phenotypes within our VANSEEK cohort. Something to keep in mind is our panel is really designed for individuals with vascular anomalies or overgrowth. So this phenotypic spectrum reflects that panel design, with AVMs being by far the most common phenotype, followed by some individuals that have a combined overgrowth and vascular malformation phenotype, and some individuals with pyogenic granulomas. And here's an example. I think this is a lesion from the thigh of one of these KRAS-altered lesions. And this lesion in particular is something that radiographically can look very concerning because it has a very solid appearance. It has very high flow. And one of the things in the radiologic differential is angiosarcoma. But when you look at it under the microscope, what you'll see are these kind of expansile lobules of capillaries that are splaying apart these skeletal muscle fibers here, together with these occasional larger vessels that are intermixed. And if you were to look at these capillaries more closely, you would see that the lining endothelial cells are plump, but they don't actually have any atypia, and there aren't many mitoses within this lesion. And so these features are actually diagnostic of an entity that's called an intramuscular fast flow vascular anomaly. And this lesion, when we sequenced it, was found to have a KRAS codon 13 variant. So intramuscular fast flow vascular anomalies are an entity that has gone by many different names and continues to have many different names in the literature. Many of the names will have some variant of intramuscular and hemangioma in them, reflecting the fact that it really has these expansile lobules of capillaries reminiscent of a benign vascular tumor. But when you sequence these, they typically have variants either in KRAS or MAP2K1, which are the same genes that are altered within arteriovenous malformations. And so the thought is that these likely represent a specific histologic manifestation of an arteriovenous malformation when it's found within the muscle. And so arteriovenous malformations are characterized by direct connections between the arterial and venous system. And because of that arterial component, these are fast flow vascular malformations. If they're superficial, you'll actually feel that they're kind of pulsatile, and they can also have a bruit. And they can be found throughout the body, including in the brain, skin, soft tissue, and occasionally in the viscera. And most of these AVMs are sporadic, but they can also arise in the setting of a few different germline predisposition syndromes, including P10 hamartoma tumor syndrome, hereditary hemorrhagic telangiectasia, and capillary malformation, arteriovenous malformation. And so our current fellow, Lisa Anderson, who will be joining us as a faculty member in July, has worked with me to help characterize the sporadic AVMs within our VANSEEK cohort. And we've identified a total of 136 individuals that have sporadic AVMs. And on the right is a pie chart depicting the genetic spectrum of these sporadic AVMs. About a quarter of them have KRAS variants, about a quarter have MAP2K1 variants, about a quarter are negative, and the remainder are pretty heterogeneous and include variants in BRAF, P10, HRAS, and RASA1. And this figure here depicts the genetic spectrum at different body sites. So the size of the pie chart is proportional to the frequency of an AVM involving that site. And then each pie chart depicts the genetic spectrum. So for example, AVMs are far more common in the limb than they are within the trunk. And when we look specifically at brain AVMs, what's interesting about brain AVMs, and others have shown as well, is that they almost exclusively have KRAS variants within them. So within our brain AVMs, there's only one that instead has a MAP2K1 variant. But when we move to other parts of the body, you can see that instead of KRAS, MAP2K1 predominates, although the other body sites are a lot more heterogeneous, and KRAS still plays a significant role. And so here is another example of a KRAS-altered lesion. This is from a teenager who had a vascular lesion on their thigh that was noted at birth and has grown over time with them. So already sounds like a malformation. But when you look at it under the microscope, there's a few interesting things going on with the histology here. Starting out with that portion that's more in the subcutis, you'll notice that there's all these kind of tiny balls of cells. And at low power, that might look kind of like that cannonball appearance that you associate with a tufted angioma. But when you look at those balls on higher power, what you'll see are very well-formed capillaries without any spindle cells, no red cell extravasation, so very different appearing on high power than a tufted angioma. And when you look at the more superficial component of this lesion within the dermis, it has these really expansile lobules of capillaries with these bigger feeding vessels. And I think taken in isolation, you could consider it to look kind of like a hemangioma or a benign vascular tumor. And this is something we see in AVMs a lot, where they don't necessarily have the histologic pattern you might expect, but instead have a little bit more of a heterogeneous appearance under the microscope and can mimic other lesions. And when we sequenced this lesion, we found that it had an duplication of amino acids 51 to 74 within KRAS. This is another example of a KRAS-altered lesion. This one's for the neuropathologists in the audience. But this is a brain lesion. And at low power, you'll notice that it has greatly increased numbers of vessels, both arteries and veins. And when you look more closely, you can see some vessels that look like this one up here, where at one side, the vessel looks well formed, but as you trace it along, you'll see that it gets this kind of profound intimal fibrosis within it. And if you were to do an elastin stain, you would see that the internal elastic lamina kind of comes and goes within that vessel. And so that's evidence of abnormal shunting. And this is a vessel that has a direct connection between an artery and vein without an intervening capillary bed. And that results in turbulent flow and that intimal fibrosis that you can see. And when we sequenced this lesion, we found a KRAS codon 12 variant. So shown here is the overall variant spectrum within arteriovenous malformations in our cohort. Down below is a schematic of the KRAS gene with the GTP binding site in dark blue and this kind of critical switch 2 domain in light blue. And the variants within KRAS AVMs kind of cluster in amino acid 12 as well as codon 61 here. And then we also have a variety of other variants that are kind of one-off and somewhat unique variants. And so how does this compare to cancer? So codon 12 is also the most commonly mutated residue in KRAS and cancer. And this is a little bit of an older paper, but I like it because they compare the mutation frequency for different KRAS variants to the biochemical activation score of that variant. And the biochemical activation score here is related to that mutated protein's ability to act as a GTPase and to activate its downstream effectors. So higher activation score is a more activating variant. And when you look, the most common variant in cancer is G12D, which is also most activating, while less activating variants are generally less common within cancer. Here I've made the same plot, but instead looking at our AVM variants. And again, the most activating variant in KRAS, this G12D variant, is also the most common within AVMs, while less activating variants like G12A are less common. And a recurrent theme as we go through some of these developmental KRAS lesions is that G12C, despite being very activating, is very uncommon in these pediatric lesions. Returning to our schematic of the gene, I've now divided it up with variants seen within brain AVMs up top and those seen in extracranial AVMs down below. And what you'll notice is that within brain AVMs specifically, there's a tendency to have those kind of hotspot oncogenic variants that you might think about in cancer, whether it's variants impacting codon 12 or codon 61. In contrast, if you look at the extracranial AVMs, they're more genetically heterogeneous and include several cases that have novel in-frame insertions, duplications, or deletions. So to summarize the role of KRAS within our AVMs, thinking first about the brain AVMs, they have a tendency to be both histologically and genetically homogeneous. The histology is generally characterized by increased arteries and veins with histologic evidence of abnormal shunting. And the genetics most commonly are hotspot oncogenic KRAS variants within those brain AVMs. In contrast, our cutaneous and soft tissue AVMs are much more histologically and genetically heterogeneous. The histologic patterns include patterns that are similar to brain AVMs with increased arteries and veins and abnormal shunting. There's also that intramuscular fast flow vascular anomaly pattern, which I showed you, and some lesions that can mimic either tufted angiomas or hemangiomas. And the genetics of these cutaneous and soft tissue AVMs is more heterogeneous and includes both our hotspot KRAS variants as well as novel KRAS variants and variants in multiple other genes, including MAP2K1. Moving on to another phenotype that we can see with KRAS, it's that of overgrowth and vascular malformations. And these patients belong to a group that was recently described a few years ago who have KRAS variants and a Klippel-Trenaunay syndrome-like phenotype. So Klippel-Trenaunay syndrome is characterized by capillary malformations together with venous or lymphatic malformations and limb overgrowth and is caused by mosaic variants in PIK3CA. But in 2021, a group of patients was described that instead have mosaic variants in KRAS and have overgrowth and vascular lymphatic malformations similar to Klippel-Trenaunay, but also have the addition of lytic bone lesions, and it seems as though they have a strong predisposition to Wilms tumor or embryonal rhabdomyosarcoma. So in this case series in particular, there were 8 individuals included, and 3 of them went on to develop cancer. The next phenotype I want to talk about with KRAS is congenital pulmonary airway malformations, and specifically the type 1 and 3 CPAMs. So congenital pulmonary airway malformations are cystic or non-cystic localized developmental anomalies of the lung. Historically, we noticed these when patients had recurrent infections, but now they're usually picked up on prenatal ultrasound and typically resected in the first year or so of life in order to prevent recurrent infections. There are many different classification systems that have been used for CPAMs, but I think the most widely adopted is the Stalker classification, which includes type 1 CPAMs, which have these very large cystic spaces, type 2 CPAMs, which generally have cysts measuring less than 1 centimeter in size, and type 3 CPAMs, which have a more solid appearance. And so this is an example of a type 1 CPAM. Grossly, you can see these large cystic spaces with relatively normal appearing intervening regions of lung. And when you look at this under the microscope, the large cysts are lined by ciliated columnar epithelium. And they frequently have architectural complexity with either papillary structures or somewhat acinar type architecture. They can also frequently have mucinous cells within them. All of these are features that we associate with a type 1 CPAM. And when this lesion was sequenced, it was found to have a KRAS codon 12 variant. This is another lung lesion, which looks very different than the prior. This one grossly looks very solid in appearance. When you look at it under the microscope, you'll see all of these tiny little irregularly shaped spaces that are lined by cuboidal epithelium. There is no normal lung mixed within this lesion, but instead what you have are these kind of broadened septa that have increased mesenchyme within them. And these are all features of a type 3 CPAM. And when this lesion was sequenced, it was also found to have a KRAS codon 12 variant. And so shown here are a few more examples of type 1 CPAMs on the left and type 3 CPAMs on the right. And in general, what's interesting is both of these lesions are caused by activating oncogenic variants in KRAS. But in general, the type 1 CPAMs are specifically caused by KRAS G12D variants. And that's our most activating KRAS variant. In contrast, type 3 CPAMs more typically have either KRAS G12V or KRAS G12R variants. And that highly activating G12D variant only accounts for 16% of the type 3 CPAMs. So it's a balancing act here between how the variant is acting and the histology that you get. Here we are returning to our graph once more of variant frequency versus biochemical activation score. And again, that most activating KRAS variant G12D is most common. And once more, G12C is less common than you would predict based upon its activation score. So to summarize the role of KRAS within these CPAMs, starting with our type 1s, those are our large cystic lesions. And the genetics are very homogeneous and typically specifically has KRAS G12D. In contrast, type 3 CPAMs are much more solid in appearance and more heterogeneous, typically have either KRAS G12V or G12R, but can also rarely have variants in other genes that activate RAS signaling. And I'd be remiss, since I talked about at the beginning, if I didn't circle back to our type 2 CPAMs. Those do not have any known genetic cause and are thought to instead arise secondary to obstruction. So why is there such a broad spectrum of phenotypes that we can see with KRAS? And this is similar to the phenotypic heterogeneity that we see in our patients with PIK3CA-related overgrowth spectrum disorder, and it's thought to be secondary to the location, activity, and timing of the variant during development. And we can look to mouse models to help provide some insight into why this happens. So shown here are 3 different mice, all of which have the same KRAS G12D variant, but expressed in different cells in the body. So for example, if KRAS G12D is expressed only within lung epithelial cells during development, you get a cystic lung lesion similar to a type 1 CPAM. In contrast, in the middle, if you instead restrict that same variant to brain endothelial cells, you get AVMs with these little knots of blue vessels. And lastly, if you restrict that same variant instead to the dorsal ectoderm, you get a skin lesion on mice, which is highly reminiscent of a nevus sebaceous. So the specific cell type that's carrying the variant is very important for the phenotype here. Additionally, turning to CPAMs, we get some insight into how activity can play a role. Within CPAMs, more activating mutations like G12D, generally resulting in more of a type 1 CPAM phenotype, while slightly less activating but still oncogenic variants like G12V more typically result in a type 3 CPAM. And then thinking about the timing of the variant, these again are all mice carrying that same KRAS G12D variant. And if it's present in the germline state, then it's embryonic lethal at about 10.5 days of development due to some defects in yolk sac vasculature. If you instead express that same variant only within the developing embryo, then the placenta can form appropriately, but it's still embryonic lethal a little bit later at 14.5 days due to congenital heart defects. And lastly, if you wait until 6.5 days of development to express that mutant KRAS specifically within the lung, then you end up with a lesion that looks like a CPAM. And so to summarize what we know about this phenotypic spectrum that we can see with oncogenic variants, both in PIK3CA and in KRAS, we think that there's probably a lot of different developmental factors at play that can result in these unique phenotypes. And that these oncogenic variants, when they occur in utero, can result in malformations instead of malignancies. But we really have a lot of work left to do to kind of fully understand the pathogenesis of these lesions and understand within any given individual which specific cells are impacted. So with the few minutes we have left, I want to move on to another topic we more commonly associate with cancer, which is a genetic complexity, but is something that we can actually see in a substantial proportion of our patients with vascular anomalies as well. So this is some work that was done by our fellow last year, Dr. Brian Wen, and he did a retrospective review of 1,300 individuals we had sequenced by VanSeq and identified 92 patients who had at least 2 different clinically reportable variants. Amongst those 92 patients, we identified 4 distinct groups or different patterns of genetic alterations. The first were individuals that had 2 variants in a single gene, where we know that 2 hits is an established mechanism of disease. The second were lesions that had at least 1 VUS. And the last 2 categories, which are the ones I find most interesting and we'll talk about today, were individuals that had multigenic variants, where we know from the literature what the implication of that is. And then a pretty substantial group of individuals that had multigenic variants where we really don't know what that means for their phenotype and patient outcome. And so shown here, this is a heat map of the different variants that we saw in individuals that had multigenic variants with a clear phenotypic significance. And each column here indicates a different patient, and each row is a gene. The colors correspond to variant allele fraction, with dark green being a variant allele fraction below 2%, and dark red being heterozygous variants. So for example, individual 66 here has a GNAQ variant with a variant allele fraction between 5 and 10%, as well as a BRAF variant with a variant allele fraction between 10 and 20%. And there are 2 subgroups here. The first were individuals who had variants within a G protein alpha subunit in conjunction with either a RAS or RAF variant. And here's an example of one such individual. They have a capillary malformation on their cheek, which I'll kind of outline here, which has been present since birth. And superimposed on that capillary malformation is a pyogenic granuloma. That pyogenic granuloma was removed. You can see the histology on the right. And when we sequenced the pyogenic granuloma, we found both a GNAQ and a BRAF variant. And the thought here is that that GNAQ variant is causative of the underlying capillary malformation, and then the BRAF variant has arisen specifically within that superimposed pyogenic granuloma. And this is a phenomenon that's been really well described. Individuals that have capillary malformations have a predisposition for developing multiple pyogenic granulomas. And when you sequence them, you will typically find both that GNAQ causing their capillary malformation, as well as either a RAF or RAS mutation causing the pyogenic granuloma. The other subgroup here were individuals who had a PIK3CA variant, together with a variant in a gene that's implicated in cerebral cavernous malformations. And cerebral cavernous malformations are slow flow brain vascular malformations that can either be unifocal or multifocal. There's a few different genetic mechanisms at play. Some of our patients will have germline variants with autosomal dominant inheritance, and that's typically caused by variants in either CRIT1, CCM2, or PDCD10. Other patients will have somatic variants instead in either one of those 3 genes, or PIK3CA or MAP3K3. And here's an example of one of those cerebral cavernous malformations. Up top, we can see a little bit of preserved, very reactive glial tissue, but most of this tissue is very hemorrhagic with a lot of dystrophic calcifications. And when you look at it closely, you can see these clusters of thin-walled back-to-back vessels. Some people call these clusters mulberries, kind of reflecting the little independent vesicles next to each other. But these are very characteristic features of a cerebral cavernous malformation. And when we sequenced this lesion, we found both a loss of function CRIT1 variant as well as a gain of function PIK3CA variant. And this is a phenomenon that's been really well described and characterized by the Marchik lab and others, wherein many cerebral cavernous malformations will have a mutation both within a kind of traditional CCM gene as well as an additional variant in either PIK3CA or MAP3K3. And they've shown through single-cell sequencing that the CRIT1 variant and PIK3CA variant is typically present within the same cell. And furthermore, when you introduce these variants into a mouse model, shown here on the right, if you have both a loss of function CRIT1 variant and a gain of function PIK3CA variant, you get these very large hemorrhagic lesions, which is much different than what you get when you have either a PIK3CA or CRIT1 variant alone. Additionally, if you move that kind of mouse model observation into a clinical observation, the patients that have CCMs with multiple variants within them generally have more severe bleeds and decreased intervals between clinically significant bleeds. So this seems to be a more severe clinical phenotype as well. Moving on now to the group of patients that I find most interesting, which are those individuals that have multiple pathogenic variants in different genes where we really don't know what the implication is for their phenotype. There's a lot of really interesting phenotypes in histology here. We're going to talk briefly about two of the patients, but this was recently accepted for publication if you all want to go dive into all of the other interesting things at play here. But one of the recurrent themes we noticed were patients who had pathogenic variants in both PIK3CA and GNAQ. Here's an example of one of those lesions. This is from the trunk of a young adult. And if we first look around the periphery, we see this very fatty lesion that has these kind of dilated, thin-walled lymphatics within it. And if you had just this peripheral part of the lesion alone, you might say, well, it's probably a lymphatic malformation. But then it also has this very discrete nodule within it, somewhat hyalinized with all of these irregularly shaped capillaries within it. If you were to take that in isolation, you might say, well, it's probably some sort of benign vascular tumor, but doesn't fit great within a specific diagnostic category. But putting it all together, this is a somewhat histologically unique lesion. And when we sequenced it, it's also genetically unique. So it has both activating PIK3CA and GNAQ variants within it. This is a lesion from an infant who had seizures and was found to have an intracranial lesion within the parietal lobe. And at low power, you'll see that it's this very hemorrhagic lesion. When you look at it more closely, there's really no normal brain tissue within this lesion, but instead it consists of all of these very irregularly shaped kind of angulated capillaries that are lined by really plump endothelial cells. There's not overt atypia within it, but it's kind of a strange looking lesion. It does not fit well within any specific diagnosis in the ISSVA classification, although there are rare case reports of what have been called intracranial hemangiomas, so you could imagine kind of lumping this in there somewhere. And when we sequenced this lesion, it also had an activating PIK3CA variant and a GNAQ variant. So very different clinical presentations, very different histology, but very similar genetics. And we really don't understand what is happening in these patients at a cellular level and the role that these different variants are playing. So to summarize what we know about genetic complexity and vascular anomalies, within our VANC cohort, about 7% of individuals have more than one clinically reportable variant. And those include some interesting groups like our individuals that have pyogenic granulomas arising in capillary malformations, or those individuals with cerebral cavernous malformations. And additionally, out of these 92 individuals, 20% of them actually have unique lesions that have multigenic variants where we don't know the role that those are playing. And those lesions often also have unique histology to go along with the unique genetics. So we have talked a lot today about the role that oncogenic variants and genetic complexity, these kind of typical cancer concepts, play in these developmental lesions. And so when do we actually cross that line, though, between malformation and malignancy? And we can turn to several of these disorders to give us a little bit of a hint. So for example, in patients that have PIK3CA-related overgrowth spectrum disorder, and specifically a very severe phenotype called Clove syndrome that's very widespread within the body, those patients have an increased risk of Wilms tumor. And we do actually screen those patients for Wilms tumor during the first few years of life. If we move instead to our KRAS-associated overgrowth and vascular malformations, that entity is really still being defined, but in the largest case series, we also see that that can serve as a cancer predisposition with patients developing either Wilms tumor or embryonal rhabdomyosarcoma. And then lastly, thinking about our KRAS-associated CPAMs, if those are either incompletely resected in infancy or resected later in childhood or in adulthood, we know that it can serve as a cancer predisposition as well. And you can have truly metastatic mucinous adenocarcinoma that arises within those CPAMs. And that's thought to arise from some of these cells you can see here. And so even within infants, you have these kind of atypical looking mucinous cells. And the thought is that over time, if the lesion lingers, then those can gain malignant potential. And so how many hits is too many? I think it is hard to say with the data that we currently have. We know from our CCM patients that at least in those individuals, if they have variants in multiple different genes, there is an increased risk of bleeding and potentially a more severe clinical phenotype. But in other lesions, we really don't know what roles the two different variants are playing or which of those variants is really dictating phenotype. And so if we return back to our schematic of how mosaic mutations can affect development, today we really focused on those variants that result in a gain of fitness. And while it's nice to kind of think of neoplasia and malformation as two separate bins that we can easily put patients into, there's probably actually a lot of lesions that kind of exist in this gray zone between these, and some that may behave more aggressively than others. And so in conclusion, I've shown you today that oncogenic variants, when they occur in utero, can result in a variety of different malformations instead of neoplasia. And the phenotypes here are very heterogeneous and likely depend on a lot of different factors, including the location, activity, and timing of the variant. And while we have some well-supported associations between multiple hits in CCMs and a more severe clinical phenotype, we don't generally know in the lesions that are more genetically complex how that's going to impact our patients. So I get to work with a really great team on these vascular anomalies, particularly our molecular diagnostics lab is fantastic. We have fabulous technicians. I also have learned a ton from my colleagues in other fields, including in hemonc, and ENT, and genetics. Vascular anomalies is a super collaborative field where I get to work with a lot of different people. I think this picture down here is a good example of that. I was in Thailand last week helping care for some patients who have vascular anomalies. And sitting here around the microscope talking about a patient are people from interventional radiology, hematology, oncology, genetics, dermatology, and pathology all working together, which is just one of the things I love about this field. And then I also wanted to briefly mention my mentor, Dr. Jenny Pogorler, who was the one who got me interested in CPAMs and somatic mosaicism way back during my first year of residency. So thank you all, and I'd be happy to take any questions.
Questions in the room? Please use a microphone so that everyone can hear you.
Very nice talking to you. So for the vascular malformations with PIK3CA mutations or mutations in the malignant pathway, what proportion of those lesions actually respond to selective inhibitors?
Yeah, that's a great question. So we have a lot of targeted therapies in our toolbox because these are oncogenes, which is great. Most of them are not specifically approved for vascular malformations. The exception is a drug called alpelisib, which is a PIK3CA inhibitor. And patients do seem to have a really great response on that. It doesn't always result in a reduction in lesion size, but when you look at quality of life measures like pain and things like that, the vast majority of patients do report an improvement in their quality of life. And we've started to move toward using some inhibitors for other parts of those pathways as well. But it's still definitely ongoing, this idea of, targeted therapy in vascular anomalies.
Do they grow back? Do they have to be on the inhibitor indefinitely?
That is a good question. We're still trying to figure that out, but in general, I think patients are counseled that they will be on the medications for a long period of time. So they're often, children who may be getting on these medicines and may potentially be on them for decades. We're still waiting to kind of figure those sorts of things out.
Great talk. So I'm trying to, so the talk was about mosaicism, right? So the animal models, were they truly mosaic, or were they just having driven expression of the mutant gene in all of the cell type, of a particular cell type, right?
Yeah. So the mouse models had, as you said, driven expression within a specific cell type or at a specific point in development. It's not really the same as, a human where the mutation randomly kind of derives during development.
So I'm not a molecular pathologist, but I was just curious about the varying allele fractions in these lesions. Is that low? Is that high? And comparing it to the animal models where presumably it's going to be like almost 100% in the lesional tissue.
Yeah, so I think the varying allele fractions can be quite low. It's much lower than we think about in cancer, where I can kind of look under the microscope and I go like, oh, those are the cancer cells, and I'm going to enrich for those and sequence them. We can't really do that with malformations. And so the varying allele fractions are quite low, and I think that reflects the fact that it's probably just a small population of cells in the lesion that maybe then has some paracrine signaling to its adjacent neighbors that results in this massive lesion forming from only a few mutated cells. But yeah, I think that's something we're still kind of, trying to wrap our heads around, is where exactly is the mutation? How can you have such a low variant allele fraction and have it actually result in such an obvious phenotype?
And then just one last question for me. I think there was a slide you also showed where you were sequencing lesional tissue and non-lesional tissue. And you mentioned that, of course, you have a lower yield in mutations in the non-lesional tissue, but what's going on there?
Yeah, so why were there still some where we had variants? Yeah, so I think that's self-selecting. So some individuals who we sequence blood for it is because they suspect that they have one of the rare germline disorders, whether that's hereditary hemorrhagic telangiectasia or CMAVM, P10 hamartoma tumor syndrome. And so it's specifically being chosen to send blood first to rule out a germline disorder. We also have some individuals where we get blood to sequence just because that's the easiest to get, and it's not expected to be positive. So that's the big group of negative blood and saliva specimens.
Other questions?
Just kind of continuing on this VAF, and how many of these have been sequenced longitudinally, and are these negatives because they were there when they formed and then have lost some of those small percentage of cells, and kind of fitting that into the clinical picture of some of these regress and some of these progress? And do you think that's driven by changes in VAF or some other thing?
No, that's a great question. So we do have some patients who have had multiple samples kind of sequenced over a period of time. And Leo's actually worked on this some during his fellowship this year. But it's probably just related to sampling for many of these. So for many patients who have a negative result, if you go on and collect another tissue sample and sequence that, they have a very high chance of having a positive result in that. So it's still around 65 to 70%, similar to that kind of first biopsy specimen. But I think there are probably some patients where either we haven't identified the gene that's causing their lesion yet, or perhaps, as you said, it might be present at a specific point in development, or maybe it just needs to be in a little specific spot in the vessel and you get this large lesion. And those are probably the patients that are recurrently negative when we sequence them over and over again.
Another question. So, I think this question of malformation versus native is obviously a huge one. And my understanding is most of those oncogenic mutations are from adult cancers. Are there pediatric cancers that are driven by some of these same mutations? And so, I guess is it that if this mutation happens early, it's always a malformation, or is it still a subset? And is there anything that we can learn from, even in that set when it's a malformation?
Yeah, no, that's a good question. I generally, thinking about PIK3CA, I don't think about that playing a big role in most pediatric cancers. RAS can certainly be activated in like embryonal rhabdomyosarcomas and some other common pediatric cancers, but most of those aren't present right at birth versus a malformation. So you can certainly try to invoke something about, a specific developmental window in utero where these variants can have a different effect.
Thank you so much.
Yeah, thank you all.